DNA polymerases. Uh, from our previous discussion, uh, we also have the idea that DNA polymerases, they are very important in the process of DNA synthesis or even in vitro DNA amplification. DNA polymerases जो हैं ये फर्स्ट टाइम 1955 में आर्थर कॉर्नबर्ग और उसके साथ कुछ और वर्कर्स थे। उन्होंने सबसे पहले इसको आइसोलेट किया इकोलाई बैक्टीरियम से और उसको नाम दिया गया डीएनए पोलीमरेज 1। Then with the passage of time, other types of DNA polymerases were also discovered. But overall, at the beginning, we can say that DNA polymerases, they are very important in the process of DNA amplification. DNA polymerases, they require uh, different factors to work, or the requirements, they may be one or two. The major requirement, first, it is the presence of DNA template. कि जो हम DNA एम्पलीफाई करना चाहते हैं, it must be available, suppose in double stranded form. तो उससे हमने जैसे देखा कि उसकी जो है डीनेचुरेशन होगी, या इन केस ऑफ इन विवो सिस्टम और बायोलॉजिकल सिस्टम the replication fork will be formed, and then DNA replication it will start in a semi-conservative manner as explained in different books of molecular biology. DNA template के अलावा जो दूसरी fundamental requirement है, it is the presence of the primers. तो naturally biological system में primers जो हैं ये होते हैं normally short segments of the DNA, or even in most of the cases they may be formed of RNA. And after binding with the complementary sequence in the template DNA, they will provide 3' hydroxyl group that will direct the synthesis of the DNA complementary strands. As I told you that according to the information that is contained in the template DNA, so it will guide the synthesis of the complementary strand. Another important factor in DNA synthesis is that what type of DNA polymerase it is being involved in DNA replication. And if we discuss this in broader perspective, that DNA polymerase, it is involved in the adding of new bases in the DNA strands when we compare it with the target DNA molecule. So according to the information, the DNA polymerase will keep on adding the nucleotide bases. After adding the nucleotide bases, it will dissociate from the DNA fragment. And after dissociation, again it will bind and continue the addition of new bases. Now it will depend that which DNA polymerase it is involved in the addition of new bases. And this ability of the DNA polymerase, that how much bases it is going to add, it is called as the fidelity of the DNA, or in other terms, it is also called the processivity of the DNA polymerase. Processivity means that how many DNA bases the DNA polymerase it is going to add before it dissociates the from the template DNA. Now, as I told you that DNA polymerases in E. coli, they may be of different types, like DNA polymerase 2 and DNA polymerase 3 that were discovered in the 1970s. और जो इसके अलावा DNA polymerase 4 and 5 you can also find, but the principal type is DNA polymerase 3 that is having high processivity and the principal enzyme in the DNA synthesis or replication in E. coli. This is the list of different enzymes of E. coli and their comparison, like DNA polymerase 1, 2, and 3, and that which is the structural gene that is involved in their synthesis, what are different subunits, what may be their molecular weight, whether they are having 3' to 5' exonuclease or endonuclease activity or proofreading activity, and what is their polymerization rate. And if we have a look on the processivity of the DNA polymerase 3, it is the most important. That is, it is having high processivity. So initially, when PCR was used for the amplification of the DNA polymerase from E. coli it was used because of its high fidelity and low error rates it mainly introduced. For example, after 1000 to 10,000 replication of the cell, the error rate it is noted one base pair. So it is a very high fidelity of the DNA polymerase of E. coli. But this is not well suited for PCR reaction that is exposed to high temperature repeatedly. So initially, when DNA polymerase from E. coli it was used, it has to be replaced or replenished after every denaturation step in the PCR procedure. So it has increased the time and even the cost of DNA amplification. So now DNA polymerase, it was worked at 37 degrees Celsius, low temperature, and E. coli DNA it is not working at high temperature. So scientists or the biologists, they find another source for DNA polymerase that was stable at high temperature. And this DNA polymerase was isolated from a bacterium that was growing in hot springs or the hydrothermal vents at the base of the ocean floors, where hot springs were common, and even in some other aquatic bodies on the land, some hot springs are present that can harbor such thermophilic bacterium, like the Thermus aquaticus. That was the microbe from where the TAC DNA polymerase was isolated. TAQ, it is from the source of the microbe from where the DNA polymerase was isolated, and it was commonly called as the TAC DNA polymerase. Now, it can be used at high temperature, but it is also having one disadvantage. A disadvantage was that it lacks 3' to 5' proofreading activity. So errors that were introduced, they were very high. So to overcome this problem, scientists look towards another hyperthermophilic microbe like the Pyrococcus furiosus. PFU DNA polymerase then was isolated from Pyrococcus furiosus and named was after the source PFU, Pyrococcus furiosus. And the advantage was that it is having high proofreading activity. And then it may be used alone or in combination with TAC DNA polymerase. So, with the passage of time, the E. coli DNA polymerases, they were replaced with more thermostable or heat-stable DNA polymerases. In the last slide, I can give the comparison or the sources of different type of thermostable DNA polymerases that were isolated from different microbes. First example is TMA. It was isolated from bacterium Thermotoga maritima, and then deep went from the ocean floors. Pyrococcus was isolated, then TLI DNA polymerase. It was from Thermococcus littoralis, and then PFU, as I told you, that Pyrococcus furiosus, and PWO DNA polymerase was isolated from Pyrococcus voicii.